Hi, this is Pratima from Planet Physiology. Welcome to the second part of Humoral Immunity. This is actually the fifth part of the series on immunity. Till now, we have studied about innate immunity, general aspects of acquired immunity, details of cell-mediated immunity, and in the previous video, we have studied formation, structure, and types of antibodies. In this part, we shall concentrate on how antibodies act against the invading organisms to destroy them, that is, the mechanism of action of antibodies. So as studied earlier, activated B lymphocytes form plasma cells which secrete different types of antibodies to fight against invading organisms. These antibodies interact with antigens to form antigen-antibody complex, which is facilitated by various types of bonds like hydrogen bond, hydrophobic bond, ionic attractions, and van der Waals forces. Once antibodies attach to the antigens, they either directly attack and destroy the antigens or they activate complement system, which in turn destroys the antigens. Let us study the various ways by which antibodies can directly act against pathogens. First, antibodies interact with antigens to form clumps. This reaction is known as agglutination reaction. Many a times, this antigen-antibody complex is very large and hence becomes insoluble in body fluids. This is called as precipitation. Sometimes antibodies just cover the toxic sites on the antigens and neutralize them so that pathogen cannot harm the tissues. This process is neutralization. And the last way is lysis, where antibodies directly attack and destroy the pathogens. Thus, antibodies inactivate pathogens directly by agglutination, precipitation, neutralization, or cell lysis. Now let us study indirect mechanism of antigen destruction by the antibodies, that is, by activation of complement system. Complement system consists of about 50 proteins in plasma belonging to globulin family. These proteins complement the effect of antibodies to destroy the pathogens, and hence it is known as complement system. About 11 complement proteins, namely C1 to C9, B and D, play very important role in destruction of pathogens. These proteins circulate in blood as inactive precursors and get activated in response to appropriate stimuli. Most of the activated complement proteins act as enzymes. Complement proteins can be activated by three different ways. When antigen-antibody interaction activates a cascade of complement proteins, it is known as classical pathway. Complement proteins are also activated by direct exposure of B and D complement proteins to antigen. It is called as alternative pathway. The third mechanism is known as lectin pathway, which is activated by activation of C3 complement protein or even directly by the antigen without presence of antibodies. Let us study the classical pathway. Classical pathway is triggered by activation of C1 complement protein by antigen-antibody interaction. C1 protein is actually a complex made of 5 subunits, namely 1 molecule of C1q, 2 molecules of C1r and 2 molecules of C1s, which is also abbreviated as C1q R square S square. Antibodies, especially IgG and IgM, have C1q binding site on their Fc region. Antigen-antibody interaction exposes this binding site and C1q immediately attaches to this antibody. It undergoes conformational change and leads to activation of C1r molecule, which in turn cleaves C1s. Thus, C1q activates classical complement cascade. That means binding of C1q with antibody initiates series of events in classical pathway of complement system. So this picture represents same. Shown in blue circle is C1q attached to the antibody. Orange colors are C1r molecules and green is C1s molecules. Now if you feel this is too complicated, just remember that antigen-antibody interaction activates C1 complement protein and activated C1 initiates complement cascade. So let us study this cascade. Activated C1 acts as enzyme to activate complement protein 4 and then complement protein 2 to form a complex called as C4b2b or just as C42b and C4a. Before we proceed further, understand the terminologies used in the cascade. Activated proteins act as enzymes and cleave another complement protein in the series. The bigger component is referred as B and the smaller as A component. Horizontal bar over the top of the product indicates that it acts as enzyme. Usually, larger part acts as enzyme to continue the cascade and the smaller component, that is the component A, acts as effector protein. Okay, let's continue. C4b2b complex acts on C3 complement protein and cleaves it into C3b and C3a proteins. C3b along with C42b splits C5 protein into C5b and C5a. C5b acts as enzyme to activate complement proteins 6 and 7 and their end products further activate complement protein 8 and 9 to form the final product called as C5b6789. This product is known as lytic complex as it is involved in cell lysis. This complex forms pores in antigens and induce osmotic lysis in the same way like your cytotoxic T cells. Complement products C3a, 4a and 5a activate mast cells and basophils to produce inflammatory response. C5a also acts as chemotactic agent and attracts WBCs at the site of infection. C3b acts as opsonin, that is, it causes opsonization of bacteria. Thus, these end products promote phagocytosis and help in removal of antigen from the body. Now coming to the alternative pathway. Alternative pathway is also known as Properdin pathway. Normally, small amount of C3 protein is spontaneously hydrolyzed, but the products are immediately inactivated in the body fluids. When the C3b is attached to the surface of pathogen, it attaches to the complement protein B to form C3bb. In presence of complement protein D, the protein B is cleaved into two subparts BB and BA. BB component remains attached to C3b to form C3b-BB complex, which acts as convertase to activate C5. So as this is another way to generate C3 convertase to activate C5, this pathway is known as alternative pathway. C3b-BB complex is stabilized by a protein called as Properdin, which greatly amplifies complement cascade and hence it is also known as Properdin pathway. Once C5 is cleaved, rest of the pathway is same as that of classical pathway. As this pathway is activated in absence of antibodies, it is the part of non-specific immunity, that is innate immunity. The third pathway is called as Lectin pathway or Mannose-binding Lectin pathway. plasma protein called Mannose-binding lectin binds to Mannose moiety on the surface of antigen. This activates serine protease similar to C1R and C1s proteins of classical pathway. So this binding of lectin with Mannose moiety splits complement protein 4 and then complement protein 2 to form C4b2b complex, which acts as classical C3 convertase. Once C3 convertase is formed, rest of the steps are same as that of classical pathway. As lectin pathway is also activated directly by antigens, that is, even in absence of antibodies, it is also the part of non-specific innate immunity. So as we have studied, various products of activated complement system promote phagocytosis and cell lysis. It has potential to be extremely damaging to the host tissue itself, and hence its activation is tightly regulated. First, the concentration of various complement control proteins in the blood is normally more than that of complement proteins themselves. Second, host cells also possess complement control proteins on their surfaces. For example, CD59 protein. These proteins prevent polymerization of C9 and hence the formation of lytic complex. Thus, host cells are protected. Next is C1 inhibitor prevents activation of C1, while decay accelerating factor DAF inhibits C3 convertase and thereby inhibits activation of classical pathway of complement system. So here we finish with the complement system. Let us quickly summarize the important points. Antibodies bind with specific antigens and can directly inactivate them by various processes like agglutination, precipitation, neutralization and cell lysis. Indirectly, antibodies can activate classical pathway of complement system by binding with C1q protein. Products of complement system either stimulate phagocytosis to clear antigens and damage cells, initiate inflammatory response to attract additional phagocytes to resolve the infection, form lytic complex to destroy antigen. Complement system also can be activated in absence of antibodies either by alternative pathway or by Mannose-binding lectin pathway. Both these pathways are part of non-specific immunity. Activation of C3 complement protein is common step in all three pathways. So that's all for this session and see you in the next video. Thank you and keep watching. If you enjoy my presentations, press the like button and share it with your friends. For more such videos, subscribe my channel and click the bell icon. Thank you for watching and see you in the next video.